continuation on our discussion on special topics in protein chemistry, we will be looking at intrinsically disordered proteins. These proteins contain intrinsically disordered regions, have specific amino acid preferences, and have specific characteristics of function and activity, which we will be discussing in this lecture. When we look at the intrinsically disordered proteins, as the name implies, they do not possess a stable three-dimensional structure. As a result, they exist in highly flexible conformations. They are functional proteins and have specific characteristics to their activity, and in some cases, the IDPs are often disordered only in localized regions, forming what are called intrinsically disordered regions, the IDRs. We look at a specific example of a dynamic view of a protein in this case, where we can see the dynamic part of the polypeptide chain, but there is a specific alpha helical component that is rigid. Maybe it is required for the recognition, but the rest of the polypeptide chain is extremely flexible in its motion. So the IDPs, as is observed, lack a hydrophobic core. The overall charge of the IDPs destabilizes the compact structure, and the interaction with the target proteins or the nucleic acids with the IDPs can often stabilize the functional conformations once they are bound to their target receptors. In intrinsically disordered proteins, they carry high densities of polar charged amino acid residues such as lysine, glutamine, serine, arginine, and glutamic acid. This is because these are interacting with the solvent more, the polypeptide chain, and there is a lack of a hydrophobic core. There is also a dominancy of the proline amino acid residues because being an amino acid, they tend to disrupt ordered structures. Also, there is a predominance of cis isomerism in the proline where we can have disordered structures as well. If we look at the distribution or the amino acid enrichment of a fully folded protein data set compared to an IDP data set, we will see the predominance, as we looked at, of proline, glutamic acid, the smaller polar ones, serine, glutamine, aspartic acid, and arginine. Whereas the hydrophobic ones are less represented. A schematic of the energy landscapes for the folded protein and an intrinsically disordered peptide indicate that because of the lack of a native structure, we can see that this corresponds to the folded peptide having a native structure, but the lack of a native structure gives it components where there are different conformations possible because of the flexibility and because of the lack of the hydrophobic core. The IDPs are involved in various aspects of cellular existence. They are involved in cell cycle and gene expression, in DNA packing, cellular transport and trafficking, communication and signaling, and regulation. When we look at these intrinsically disordered proteins, because of their lack of structure, they are also found in common pathologies such as cancer, neurodegenerative diseases like Alzheimer's disease and Parkinson's disease, in addition to the Creutzfeldt-Jakob disease. The methods of determination of structures from intrinsically disordered proteins indicates that it is not possible by X-ray crystallography because they are not ordered. They do not we cannot get a crystal of these proteins, but they can be looked at from a spectroscopic point of view from NMR, CD, or single molecule FRET, where indications about elements of secondary structure, if present, may be obtained, and also high-speed AFM. Raman optical activity, there can be time-resolved small angle X-ray scattering, and computational methods that go for predictions of IDPs. But given their flexibility, these are difficult to characterize. The specific functions for the IDRs or the IDPs is they can be targets for post-translational modifications. As a result, they are involved in protein-protein and protein-nucleic acid interactions. They can store small ligands, and they can be chaperones where they assist the folding of proteins and RNA molecules given their flexibility. The molecular recognition that occurs in IDPs is that while they function or while they are active, they fold during that time. So in that case, they can act as assemblers, where they act as scaffolds in the assembly of large protein complexes, such as the ribosome or cytoskeleton or chromatin. They can also act as effectors, where they regulate the activity of other proteins or other regions of the same protein. And this is all because they do not have a rigid structure, and they are flexible in their way their polypeptide chain can function. This means there is a large amount of entropic chain activity because of the flexibility that is permitted or present in the intrinsically disordered proteins. So what happens in this case, there is a force that opposes the structural changes or affects the orientation or localization of attached domains within the protein. As a result, they do not fold during their activity, but they can also be linkers, where they could link protein regions, that is specific domains. And in this case, what happens, they acquire different orientations with respect to each other. The advantages in the context of protein-protein interactions in comparison with regular structured proteins and domains is their ability to interact with multiple partners. This promiscuity sometimes helps in the multitude of partners that they can interact with. They can fold into different conformations depending upon the partner, and they can undergo disordered to ordered transitions based on their functional activity. So depending upon the requirement, they can adapt to the specific situation given their flexibility. If we look at the characteristics of these IDPs and IDRs, they use often a smaller protein size to provide for the same interface area, because we do not have a specific scaffold of another protein that has to have a specific protein-protein interface. Since these portions are flexible, they can form an interface using a smaller protein size. They can also wrap around the target to overcome steric interactions. Because of their flexibility and lack of structure, they have the ability to interact in a different fashion. Also, they can be complementary binding surfaces. There are unfavorable conformation entropy changes, high specificity, but low affinity. So when we look at the protein regions, there is a residual structure often, and these includes mainly the PP2 type of helices that we looked at in protein-protein interactions, with 10 to 20% of residues involved in this fashion. And the binding and folding occurs mainly via linear motifs. These are molecular recognition features or molecular recognition elements, because we realize that if there is a protein-protein interaction or a protein polypeptide interaction to occur, it is essential that there is molecular recognition. This can occur through specific features on the protein or specific elements of the protein. The morphs or the mores, they are longer in size in having 10 to 70 amino acid residues, whereas there are slims that are conserved short linear motifs that are relatively shorter in size from 3 to 10 amino acids, but these are required for the recognition of the flexible IDP with the target receptor. So the mores and the morphs, the molecular recognition elements and features, they tend to be amphipathic in nature, and the short linear motifs can serve as ligand binding sites. We have seen examples of this where we have the tyrosine containing motif that is recognized by the SH2 domains, and the PXXP, the polyproline type of motifs, recognized by the SH3 domains. They are relatively shorter than the molecular recognition elements and features. If we go back to look at the residue preferences, therefore, we found that proline is overrepresented, prevents it from forming stable secondary structures, because this would result in a lack of a hydrophobic core of a specific structure being formed, where proline, as we know, is disruptive in nature in that sense. The polar residue overrepresentation prevents the stable non-polar core in this case. So if we look at the cysteines are underrepresented. This can be understood because we do not want the disulfide bonds in this case to stabilize the tertiary structure. The short linear motifs have low affinity binding, resulting in fast exchanges, and they are found preferably in post-translational modification sites. There is low evolutionary conservation because of the lack of buried residues, the lack of a hydrophobic core, and this feature is used in many prediction tools to determine the structures of IDPs. So if we look at the structure-function relationship in IDPs, the IDPs bind their ligands with higher specificity than globular proteins. The recognition sites bind in a fashion that show higher specificity because they can adapt to the specific target receptor, and their higher binding flexibility does not contradict the specificity. Under the same conditions, IDPs are able to bind to a single target protein. As a result of this flexibility, there is a plasticity associated. This plasticity means there is a higher potential to bind other proteins when there is a change in cellular condition. So the fact that a single IDP could bind to a number of proteins is because of their flexibility, their lack of rigidity, and a change of conditions could result in a plasticity where they could bind in a different fashion with different receptors, given that the conditions are changed. So if we look at a conformational flexibility where we have a plasticity of binding to different proteins under different conditions, then if we look at a specific hypoxia inducible factor shown in red, this is bound to different targets. So in this case, we see on the left, we see a predominantly high alpha helical structure, where here there is a predominant polypeptide chain that shows an unstructured region with a short region showing an alpha helix. So it is the same sequence that is interacting with different proteins under different conditions and adapting to the binding based on the conformational flexibility. So if we look at the binding affinity, we have a delta G bind that is equal to the delta H bind minus the T delta S bind. Now, given that this is a disordered structure and when binding does occur, The binding and folding in this case is coupled. So there is a larger delta S binding compared to globular proteins, but the binding is weaker in this sense. However, IDPs have a higher partner exchange rate. Now, this is useful for signaling proteins. IDPs cannot use the delta H bind to drive catalysis because the enzymes are not intrinsically disordered. They have a specific structural condition that they adopt. But the non-polar binding surfaces can provide a larger delta H bind that actually compensates for the entropy loss. So when we are looking at the non-polar binding surfaces in this case, they provide a delta H bind that gives a rigidity to a certain portion where the interaction is occurring. What we see here is the solution structure of the unstructured protein with a zinc finger. As we see the flexible regions that are moving around in the way that they could interact with other target receptors, the zinc binding site, on the other hand, is regulated, is coordinated with the specific residues that belong to secondary structural elements. So the fact that this part is rigid compared to the rest of the protein is important in its structure function relationships. If we look at the binding folding kinetics in these IDPs, there are weak interactions of these partially folded IDPs, but what can happen is it is possible for them to sample many conformations given that they do not have a rigid three-dimensional structure. As a result, the specific binding site can be searched for and found while the folding is occurring. This gives a high affinity conformation with stronger binding possible because it can sample different types of conformations. This then enhances the folding speed as well. If we look at the binding specificity, for example, in the pp2 helix with unpaired backbone polar groups, there is a possibility of additional hydrogen bonding. The unsatisfied internal hydrogen bonds in the secondary structure, where the folding is partial because the pp2 helices do not have any internal hydrogen bonds. As a result, there are more hydrogen bonds available for ligand binding, resulting in specific binding that gives us a specific structure function relationship in accordance with the binding characteristics. If we look at the regulation via post-translational modifications, the flexibility again comes into the picture in these intrinsically disordered regions of these proteins. So the disordered structure results in an extended chain. As a result of this, there is high accessibility to modifying enzymes. So whether these enzymes are involved in phosphorylation, acetylation, ubiquitination, hydroxylation, or even methylation, there is high accessibility possible due to the extended chain conformation of these specific IDPs. The short linear motifs in this case, they have specific determinants that are recognized by the PTM enzymes in resulting in the specific modifications that occur. If we look at the short linear motifs for a specific type of protein, in this case, the bromo domain and the PhD finger that contains a protein transcription factor, the BPTF, this is involved in chromatin remodeling and is therefore a potential anti-cancer target. In this case, a short linear motif binds to the BPTF binding site, and this is a methylated lysine that is the SLIM, that is a histone derived peptide. So the methylation of the lysine 4 allows it to fit very snugly into the BPTF binding site that includes 3 tyrosine residues and 1 tryptophan. So further design of inhibitors of the BPTF binding site can be developed based on this knowledge. The IDPs and IDPRs are variable in sequence and structure and therefore are not homogeneous. They possess a complex architecture that is indicative of independently foldable units of a protein that could form secondary structural units at some portions where we saw for the zinc binding domain. They could have disordered regions that can partially fold to facilitate interactions with target receptors, like we saw for the DNA binding or for any flexible binding where the binding folding kinetics is of importance. There can also be flexible regions that can undergo transitions to ordered regions in order for them to be functional. So this transition from a disorder to an order region, again, looks at entropy and enthalpic factors that are interesting for the binding folding characteristics of the IDPs. If we look at a schematic representation of amyloid formation, we have an unfolded protein. The amyloid formation is observed in intrinsically disordered proteins. An unfolded protein can go to an intermediate folding state that then desirably would go to a native state. However, this intermediate may also form non-amyloidogenic oligomers, as a result of which there could be structural changes, and these structural changes could result in these specific amyloid fibrils that are the cause of many diseases that result in amyloidosis. For example, the tau protein, this is caused by intracellular tangles of a polymerized form of a microtubule accessory protein, the tau protein, and the amyloid beta. The tau protein is mostly found in the brain. In this case, it is 3 to 4 fold more hyperphosphorylated than the normal tau protein in case of the disease. This hyperphosphorylated tau is then polymerized into paired helical filaments and straight filaments that form these neurofibrillary tangles that are the hallmark of Alzheimer's disease. So when we look at Alzheimer's disease in these IDP associated diseases, it is a neurodegenerative condition that is found mainly in elderly patients, 10%, approximately 10% over 65 years and 50% over 85 years. And the structural aspects that we see, the variations in the structure that result finally in these amyloid beta peptides that deposit in the brain as amyloid plaques. So this is caused by the accumulation and the deposition of the amyloid beta protein. This amyloid beta protein is intrinsically disordered. So the cause of Alzheimer's disease is histologically characterized by the presence of these neurofibrillary tangles formed by the tau proteins and the amyloid plaques formed by the extracellular aggregates of the A beta peptide. So an understanding of the IDPs is important. In summary, the intrinsic disorder is a unique structural feature that facilitates the participation of these IDPs in a multitude of interaction possibilities. As we have seen, these interaction possibilities are such that they have unique binding folding characteristics, and because of their flexibility, they can adapt to different conformations depending on different conditions. However, this also leads to disease. As such, they are associated with many diseases such as amyloidosis, cancer, cardiovascular disease, diabetes, and neurodegenerative diseases. The understanding of these intrinsically disordered regions, the intrinsically disordered proteins, their amino acid preferences, and their structure function relationships will lead us into a further understanding of the IDPs and their unique binding folding kinetics and characteristics. These are the references. Thank you.